Knowledge of biodiversity is vital to understand ecological functions of oil palm plantations. We measure the changes in diversity of major taxonomic groups involved in diverse ecological processes. For example, at the canopy level, we record the occurrence of insectivorous bats, which serve ecological roles as predators of night-flying insects, controlling pests, and in pollination and seed dispersal. We also investigate the arthropod diversity. Here you see pictures of some insect arthropods which provide ecological roles in pollination and pest control. Below ground, we quantify soil animals, for example, columbola, that play a key role in plant litter decomposition. Protozoa, which are involved in nutrient cycling. Here are pictures of amoebae, which have a significant role as bacterivores. At the root level, we investigate the colonization rate and diversity of fungal endophytes. Among them, the mycorrhizal fungi, which contribute to plant nutrient uptake and are important components of soil food webs. Soil microbial diversity is investigated using DNA-based molecular identification by high-throughput sequencing. Soil samples are immediately frozen in the field and kept frozen during the transport to the University of Göttingen, where nucleic acids are extracted and the target DNA region amplified and subsequently sequenced in order to obtain the taxonomic assignment and the soil bacterial and fungal community composition. For environmental processes, we quantify nutrient leaching losses. In each replicate plot, we installed 6 lysimeters, 2 at the palm circle where fertilizers are applied and weeded 4 times a year, 2 at the interrow where there is no fertilization and weeded 2 times a year, and 2 in the frond stacked area where Sinicet fronds are piled. Suction cup lysimeters are inserted into the soil at a depth of 1.5 meters below the rooting zone. Tubes connecting from the lysimeters to the collection bottles are protected against animals with plastic tubes and buried in the soil. The collection bottles are kept in the dark to avoid algal growth by putting them inside buckets. Leaching water is sampled by applying a pressure of 40 kPa to the lysimeters. Water sampling is done bi-weekly during the rainy season and monthly during the dry season. We also quantified soil greenhouse gas fluxes, nitrous oxide, methane, and carbon dioxide. In each replicate plot, we measured these gas fluxes from the palm circle, frond stacked area, and interrow. We used a vented static chamber. The chamber bases are left permanently, and these are covered for 30 minutes on each sampling day. And during these 30 minutes, 4 samples are taken and stored in pre-evacuated exotainers. Measurements are conducted monthly. Follow-on measurements are conducted more intensively during 6 weeks following fertilizer applications. On each sampling day, mineral nitrogen and carbon are extracted in the field to avoid errors from soil storage. Further weighing, shaking, filtering, and water content determination are done at the University of Jambi. Frozen extracts and the gas samples are transported to the University of Göttingen in Germany for analysis. For cost-benefit analysis, one of the costs is fertilizers, and these data are gathered from the actual market price. Another is labor costs, and the working hours and actual salary with benefits are recorded.